আসসালামু আলাইকুম ওয়া রাহমাতুল্লাহ। আমরা আজকে হাইপার সেনসিটিভিটি টাইপ 2 এখানে 2 to 4 এই দুটো ডিসকাস করব ইনশাআল্লাহ। রাব্বি জিদনি ইলমা রাব্বি ইশরাহ লি সাদরি ওয়া ইয়াসসির লি আমরি ওয়াহলুল উকদাতাম মিন লিসানি। হাইপার সেনসিটিভিটি টাইপ 2 to 4। হাইপার সেনসিটিভিটি টাইপ 2 এটা আরেকটা নাম হলো সাইটোটক্সিক হাইপারসেনসিটিভিটি। নাম দেখে বোঝা যায় যে এখানে সাইটোটক্সিসিটি হয় এই হাইপারসেনসিটিভিটি। In type 2 hypersensitivity, the antigens are usually present on the cell surface. They are mediated by IgG and IgM also. An immune process involved in type 2 hypersensitivity includes classical complement pathway, ADCC via NK cell or eosinophil, and opsonization, 3 pathways. Many autoimmune diseases result from type 2 hypersensitivity. A classical example is rheumatic fever. Examples of type 2 hypersensitivity diseases are transfusion reaction, Erythroblastosis fetalis, autoimmune hemolytic anemia, myasthenia gravis, Graves' disease, hemolytic anemia, rheumatoid arthritis. The mechanisms involved in type 2 hypersensitivity, one of the mechanisms is opsonization. You see, this is a IgG mediated opsonization. IgG present on the cell surface. So cell surface antigen is present on cell surface. IgG bind, then Fc portion free. In that Fc portion, phagocytic cell came and releases, discharge their lysozyme, resulting in cytotoxicity. So this is one mechanism. You can see again. Second mechanism is complement mediated lysis. You see, 2 immunoglobulin G bound to the antigen, then classical pathway starting from complement 1 to 9, membrane attack complex forms, and thereby it leads to cell damage. So this is the second mechanism of type 2 hypersensitivity. ক্লাস নিচ্ছে ঠিক আছে, তারপর পরে রাখবো। রাখো। Type 2 hypersensitivity, another mechanism is antibody dependent cell mediated lysis. This is a classical example in autoimmune hemolytic anemia. There is target cell, say RBC, the antibody bound there, and following binding with the implement with the antibody, complements are activated, and there is lysis, either by opsonization via C3b or by membrane attack complex. This is the animation of antibody dependent cell mediated cytotoxicity. You can see the name is, the antibodies involved are immunoglobulin G, cells involved are natural killer cell, and following the binding of antibody with the antigen, the natural killer cell come with the Fc receptor bound with the immunoglobulin G Fc, and there is discharge of perforin and granzyme, which results in cell lysis. So third mechanism, one is opsonization, another one is complement mediated lysis, and third one is antibody dependent cell mediated cytotoxicity. This is a diagrammatic examination of Erythroblastosis fetalis. It is self-explanatory. During the first pregnancy, the RBC with antigen if the mother is negative and the fetus is positive, so this antibody, this antigen on the positive on the RBC that stimulates the mother to form antibody against RBC, and during the second pregnancy, the IgG antibody crosses the placenta and causes erythroblastosis fetalis. This can be prevented by treating the mother with ROGAM, that is antibody against the RBC, which prevents B cell activation and memory cell formation. So no IgG antibody will be produced that can destroy the fetus. This is another the example of type 2 hypersensitivity, where the antibody are formed against the acetylcholine receptor. So acetylcholine cannot reach the acetylcholine receptor. Rather, the antibody against the acetylcholine receptor, they bind with the acetylcholine receptor and results in disturbance or impairment of neuromuscular transmission, that is myasthenia gravis. But type 5 hypersensitivity, that is stimulated opposite, where the antibodies are formed against TSH receptor and they cause stimulation of thyroid gland, forming more T3, T4. Now, type 3 hypersensitivity. Type 3 hypersensitivity, also known as immune complex mediated hypersensitivity. Here also immunoglobulin G, IgM, and complements are involved. So in both type 2 and type 3, immunoglobulin G, M, and complements are involved. And the process involved are two processes: classical complement pathway and phagocytic cell activity. Immune complex, you can see the animation here. Say, when large quantities of soluble antigen antibody complexes are formed in the blood, they are not completely removed by the macrophages. These antigen antibody complexes, they lodge in between endothelial cells and basement membrane. They lodge there. These antibody complex activate the classical complement pathway. Then leukocytes will come here. Leukocytes discharge their killing agents and promotes the massive inflammation. This leads to tissue death and hemorrhage. There are two types of immune complex mediated hypersensitivity. One is Arthur's reaction, when the reaction is localized in excess of antibody. The proportion of antibody is more than the antigen, so the immune complexes are formed at the site of formation and cause localized damage. This is Arthur's reaction. And serum sickness, the antibody proportion is less, so soluble small antigen antibody complexes are formed and they deposit in the filtering surfaces like glomerular basement membrane. There are three phases. In the first phase, immune complexes are formed and they circulate. These antigen antibody complexes, they are deposited in the filtering surfaces. Then they attract the leukocytes, complements, there is platelet aggregation. All these things cause damage. And classical example is this glomerulonephritis. You see the diagram. Antigen antibody complexes, they activate the complement. There is platelet aggregation as well as activation of Hessman factor. This too leads to vasodilatation and edema, and complement activation leads to chemotactic factor that attracts neutrophil. This neutrophil, they release the lysosomal enzyme, causes necrosis, and the platelet aggregation and the coagulation pathway activation results in microthrombi formation, ischemia. That also leads to necrosis, and all these are the consequences of antigen antibody complexes. Type 4 hypersensitivity, also known as delayed type of hypersensitivity or cell-mediated hypersensitivity. This is the only hypersensitivity where no antibody is involved. Here, soluble or cell-associated antigens are involved. And when the antigens, they are processed by the dendritic cell, they stimulate the T cell, T helper cell. T helper cell produces cytokine, then they activates the macrophage. Cellular infiltrate is there, including polymorphonuclear leukocyte and resulting in edema. There are three types of type 4 hypersensitivity: delayed type, contact hypersensitivity, and gluten sensitive. Delayed type of hypersensitivity, usually protein antigens are involved. Classical example is mycobacterial proteins, that is tuberculin test. There is local swelling in tuberculin test, what happens? Erythrema, induration, there is cellular infiltrate. But in contact hypersensitivity, instead of protein antigen, haptens are involved and also small metal ion. This leads to local epidermal reaction, that is erythrema and cellular infiltrate, no induration. And in gluten hyperenteropathy, celiac disease, the gliadin antigen is involved and there is villous atrophy in the small bowel resulting in malabsorption. So, intracellular pathogens and contact antigens that induce delayed type of hypersensitivity. You know, intracellular bacteria, Mycobacterium tuberculosis, lepri, Listeria, monocytogenes, Brucella abortus, intracellular fungi, Pneumocystis carini, Candida albicans, Histoplasma capsulatum, Cryptococcus neoformans, and intracellular parasite, that is Leishmania. Examples of virals are herpes simplex virus, measles virus, and there are some small metallic poison like hair dye, nickel salt, poison ivy, poison oak, etc. And there are three phases: sensitization phase, effector phase. In two phases. In sensitization phase, intracellular bacteria or intracellular microbe, they are presented to the T helper cell. T helper cell becomes Th1, and Th1 they also recruit the CD8 occasionally, and they secrete cytokines and sometimes macrophages. In the effector phase, the sensitized Th1 cell, they secrete the gamma interferon and TNF factor B, and they activate the macrophages. These macrophages, they increases class 2 MHC molecule, TNF receptor, increase oxygen radical and nitric oxide, and Th1 secretion include gamma interferon, tumor necrosis factor B, interleukin 2, interleukin 3, GM, CSF, chemokines, and all this causes this delayed type of contact hypersensitivity and classical formation of tubercle. So you can see this picture, poison oak, an example when it comes in contact with the skin. This skin protein with cell protein, this is a hapten, so they are processed and presented to the T helper cell. It becomes sensitized Th1. Th1 secretes gamma interferon, myeloperoxide is 1, and they activate the tissue macrophage and they attract the monocytes, and all these things causes contact dermatitis. This is the positive tuberculin test. This is delayed type of classical example. There is induration, contact dermatitis, there is reaction in the epidermis, there is no induration. This is maybe Th1 or CTL mediated hypersensitivity. Patch test is used to diagnose delayed allergic reactions such as contact dermatitis, in which the contact allergens are taped on the skin underneath a special aluminum disc and keeping them for 48 hours. It can test allergy for rubber, nickel, etc. You see, type 1 hypersensitivity, there are similar type of allergen tests. It takes only few seconds to minutes. But PES test is kept for 48 hours. Diseases of type 4 hypersensitivity includes chronic active hepatitis, contact dermatitis, graft rejection, and inflammatory bowel disease. And this is a schematic illustration of events that give rise to formation of granuloma in type 4 hypersensitivity. And there is role of Th1 and activated macrophages. There is no role of any antibody. This Th1 and macrophage. Thank you very much.